Welcome to this lecture series on this eukaryotic gene expression, basics and benefits. The last three classes we have been discussing how regulation of gene expression is important in regulating the embryonic development. We started discussion about early embryonic development, how right from the stage of oocyte the regulation of embryonic development begins. Even at the stage of oocyte development, as the oocyte is being formed, the nerve cells actually start pushing some messenger RNAs into the oocyte cytoplasm, and these messenger RNAs, known as the maternal messenger RNAs, get deposited inside the oocyte cytoplasm in different regions. Some get distributed in the anterior region, some get distributed in the posterior region, but they don't get translated. And then after fertilization, these maternal messenger RNAs get translated, and then these proteins, which most of them which are transcription factors, now induce the expression of what are called as the zygotic genes. And we discussed in the using Drosophila as an example, we discussed about what are called as gap genes, pair role genes, segmentation genes. And the take home message from those early lectures is that spatial and temporal expression of these genes play a very important role in the normal embryonic development. So what we will do in this lecture is to just recapitulate some of the observations we made in the earlier two lectures, and then proceed to a very important part of development regulation, namely homeotic genes, because these homeotic genes are the ones which are ultimately responsible for giving identity to the various segments of the Drosophila and also for the formation of various organs. So we will initially discuss how homeotic genes are important, what happens when you have mutations in these homeotic genes, not only in Drosophila but also in vertebrates like mouse and humans, and how important are these homeotic genes and how the expression of these homeotic genes are actually regulated and where they are expressed, how they are regulated, and how do they bring about all these various aspects of developmental regulation. So this cartoon just shows some of the very popular homeotic mutations which were originally identified in Drosophila. Like if you mutate one of the homeotic genes in Drosophila called as ultrabithorax, you can convert, instead of the fly having one pair of wings, it will now start having two pairs of wings. Similarly, if you have a mutation in another homeotic genes called antennapedia, instead of antenna coming from the head, you will actually have legs coming from the head, clearly indicating that these genes are master regulators or master control genes, and if you meddle with them, the entire organ identity can be changed. So let us now try to understand in detail how these homeotic genes function and how these homeotic genes are activated and what are the target genes for these various homeotic genes. So this slide just recapitulates some of the very important observations we have made in the last three classes. Like I said in the beginning, the regulation of embryonic development begins right at the stage of oocyte formation or the oocyte development. Even as the oocyte is being formed, the nerve cells in the ovary, through what are called as cytoplasmic bridges, actually synthesize messenger RNAs and transport these messenger RNAs through the cytoplasmic bridges into the oocyte cytoplasm. And these maternal RNAs, known as the maternal RNAs, maternal messenger RNAs, they don't get translated till fertilization, but they get deposited in different regions of the oocyte. For example, an RNA called bicoid mRNA gets deposited in the anterior region of the developing oocyte, whereas another mRNA called the bicoid mRNA gets deposited in the another mRNA called the nanos mRNA gets deposited in the posterior region of the oocyte. So after fertilization, when these messenger RNAs get translated, the bicoid protein finally gets deposited in the anterior region, whereas the nanos protein gets deposited in the posterior region. And so differential localization of this maternal effect gene products in the oocyte cytoplasm before fertilization plays a very important role in triggering the expression of some of the early zygotic genes during embryonic development. So the bicoid and nanos, which are just two examples I had mentioned, there are many such maternal effect genes, we will not go into the details. The bicoid and nanos, for example, have opposite effects on a gene called hunchback. Hunchback is a zygotic gene, is an RNA which gets expressed now when that bicoid transcription factor goes and binds the promoter of the hunchback gene gets expressed and the hunchback protein is made. So bicoid is the transcription factor that activates hunchback. But since the bicoid is present primarily in the anterior 2/3 of the embryo, the hunchback gets activated only in the hunchback protein is made in large amounts in the anterior region, but the hunchback protein that is even made in the posterior region does not get translated because the nanose protein, which is now present in large amounts, goes and binds to the hunchback messenger RNA and prevents its translation. So, as a result, the hunchback protein can act only in the anterior 2/3 of the embryo, whereas the nanose prevents the activity of hunchback in the posterior region. So, the nanose inhibits the translation of hunchback messenger RNAs. The combined effect of bicoid and nanose results in the production of high levels of hunchback protein at the anterior region of the embryo and low levels towards the posterior end. So, these proteins called bicoid, nanose, hunchback, they are all called as morphogens, and you can see they are all present in a gradient, either in the anterior-posterior gradient, or if you take proteins like dorsal and ventral, they are present in the dorso-ventral gradient, and so on and so forth. So, by creating these kinds of a morphogen gradients, differential gene expression is brought about during early embryonic development. So, the combined concentration gradients of bicoid, nanose and hunchback sequentially activate or inhibit other morphogens genes in specific regions of the embryo. So, since hunchback is present in high concentration anterior region, it will now activate only those genes in the anterior region of the embryo. So, the genes which have to be activated by hunchback will not be activated in the posterior region of the embryo, because nano prevents the translation of hunchback, and therefore, hunchback will not be able to activate those genes in the posterior region. So, target genes of hunchback will be activated only in the anterior region of the embryo, but not in the posterior region of embryo, and this will have tremendous implications in the further development of the embryo. So, in addition to discussing some of these maternal effect genes like nanose, dorsal, bicoid and so on and so forth, we also discussed how these molecules also influence various signal transduction pathways. As I said, this is not a lecture series on developmental biology, so we cannot cover the entire developmental programs, but I gave a few examples of very important signal transduction pathways that operate during the embryonic development of Drosophila as well as vertebrate development. Some of them which we discussed in detail are what is called the hedgehog signal transduction pathway, engrailed, notch, Wnt and so on and so forth, and we discussed in detail how when you have mutations in this signal transduction pathway, it can lead to abnormal embryonic development and even diseases, and many of these pathways are also very important in the adult stages as well, and if you have mutations in some of these pathways, it can lead to genetic disorders as well as it can lead to cancer. Like Wnt pathway is very important, and if you have mutations in the Wnt components of the Wnt signaling, you can get colon cancer. So, all these signal transduction pathways are important not only during embryonic development, but they also play a very important role during the adult life as well. So, in addition to some of the pathways that we discussed, there are also many other important signaling pathways that play a very important role in embryonic development. For example, the fibroblast growth factor, the TGF beta family of proteins, platelet derived growth factor, Ephrin. There are so many signaling molecules which play a very important role in activating the expression of a number of target genes, both in spatially and temporally, that means in space as well as time, and as a result of all these signaling events, the embryonic development proceeds smoothly, and from a single cell embryo you get an adult finally. So, just to summarize the key points of the early embryonic development we have studied so far, there is the two important things that are important for the early embryonic development is the development of an anterior-posterior axis and a dorso-ventral axis. That is, anterior and posterior end the embryo has to know which is the anterior end, which is the posterior end, which is the dorsal end, which is the ventral end. So, as far as the anterior posterior axis development is concerned, the bicoid protein plays a very important role. So, the bicoid protein gradient switches the hunchback at a very high concentration in the anterior region of the embryo, and hunchback in turn now activates or represses gap genes like Kruppel, GNPS, JAINT and so on and so forth, and then these gap gene products and gap genes interact and sharpen the expression boundaries, and then the axis now gets divided into unique domains containing different combinations of this transcription factor. So, the bicoid protein plays a very important role in this anterior posterior gradient. Similarly, the dorsal ventral gradient, you need to know which is the dorsal, that is the back, and which is the belly, which is the ventral region. So, these axes are to be specified during early embryonic development, then only you get a normal organism from the embryo. The dorsoventral axis, again the gradient of the intranuclear dorsal protein plays a very important role, and the ventral activation of twist, snail and repression of decapentaplegic are very important. These are all targets of the dorsal protein. The protein called as decapentaplegic gets expressed only dorsally, and the gradient of the decapentaplegic activity patterns the dorsal region, and the dorsoventral axis is divided into prospective mesodome, neurectodome, epidermis, and so on and so forth, because of the activity of all these maternal genes, gap genes, and so on and so forth. So, once the gap gene products are formed, the gap genes now activate what are called as the pair rule genes and as well as segmentation genes, and production of local combinations of the gap gene transcription factors activate what are called as the pair rule genes in 7 transverse stripes along the anterior-posterior axis of the embryo, and the result of this pair rule gene expression is the formation of what are called as 14 para segments, each pair rule gene being expressed in alternate para segments. So, these are some of the important points that we discussed in the last 2 lectures. So, by the action of these maternal, gap and pair role genes, the Drosophila, which was a single cell embryo at the beginning, now becomes a multicellular embryo, and it gets divided into about 14 para segments. So, these are known as PS1 to PS14. Now, in the adult fly, these 14 para segments are responsible for the formation of 3 head, 3 thoracic, and 8 abdominal segments. So, the first 3 para segments give rise to the head, the next 3 give rise to the thoracic segments, and the remaining give rise to the abdomen region of the embryo. A very important point, the relationship between the para segments and the actual segments is that although the number of para segments and number of segments number is similar, their arrangement is little bit peculiar. So, although there are similar number of segments and para segments, they are slightly shifted relative to one another. In the thorax and abdomen, this shift is approximately half a segment. You can see here, if you now for example take para segment 6 as an example, the para segment 6 here comprises of the posterior segment of segment T3 and anterior segment of A1. That means the last thoracic, the para segment 6 consists of, this is T3, the thoracic segment 3 and abdominal segment 1. And you can see the para segment 6 actually comprises of the posterior region of the thoracic segment 3 and anterior region of abdominal segment 1. This is true for all the para segments. If you now take for example para segment 9, it consists of posterior regions of the previous segment and anterior region of the next segment. Similarly, para segment 12 consists of the posterior region of A6 and the anterior region of A7. So, there is a very unique arrangement between the para segment and the segments during the development. So, a para segment therefore comprises the posterior half of one segment and anterior half of the next segment. This plays a very important role in the development of the embryo. So, now let us now focus on, as a result of all the expression of these gap genes, pair rule genes, segmentation genes, now the embryo has to now become an adult. This is where the homeotic genes plays a very important role, because it is the homeotic genes which gives the identity to each segment. So, let us now try to understand what are these homeotic genes and how do they function. In Drosophila, there are about 8 homeotic genes which regulate the identity of regions within the adult embryo. And these 8 homeotic genes are clustered in 2 complexes. One is called as the antennapedia complex, another is called as the bithorax complex. The antennapedia complex consists of homeotic genes known as labial, protocypedia, deformed, sex chromes reduced, and antennapedia. These are the major homeotic genes of the antennapedia complex. The bithorax complex consists of 3 homeotic genes, the ultrabithorax, abdominal A and abdominal B. And as you can see in this picture, these homeotic genes are expressed in different regions of the embryo, with the bithorax complex getting expressed in the posterior region and the antennapedia complex getting expressed in the anterior region, and so on and so forth. So, these homeotic gene products are nothing but they are transcription factors. All these homeotic genes contain a stretch of DNA sequence known as the homeobox sequence, and on transcription and translation, this homeobox sequence codes for a 60 amino acid domain which is known as the homeodomain, which is nothing but it can bind to specific DNA sequences. So, the sequence specific DNA binding activity of the homeodomain proteins is because of the presence of the homeodomain. We had discussed in detail this homeodomain structure when we discussed some of the structures of various DNA binding proteins in the previous classes, where we discussed about leucine zipper, zinc finger, helix turn, helix motif, and so on and so forth. The homeodomain is nothing but a helix turn, helix motif. It consists of 3 alpha helices and this is the sequence of the homeodomain region of a homeobox protein known as the Antennapedia. And this homeobox sequence is very important because it is this homeobox which now binds to specific sequences of the promoter regions of the target genes, and therefore are directly responsible for activation of various target genes during embryonic development. This just shows you the multiple alignment of the various homeodomain regions of all the homeotic genes of Drosophila. Just to tell you that this homeobox region has many conserved amino acids, especially the helix 1, helix 2 and helix 3 region, which is involved in DNA binding, is highly conserved because they form the helix turn helix motif of all these homeodomain transcription factors, and therefore is responsible for sequence specific DNA binding, and therefore is highly conserved in all the homeotic gene products of the Drosophila. So, what are these homeotic genes? What do they do? Homeotic genes are nothing but they act as selectors, that is, they activate the construction of segment specific traits like wings, legs etc. So, the homeotic genes which are expressed in the thoracic region tells the segment that it is from these segments the wings have to come or legs have to come, and the homeotic genes which are expressed in the head region are responsible for the development of antenna and eyes in the head region, and so on and so forth. So, basically the development of segment specific traits like the development of eyes or antenna from the head or development of legs and wings from the thoracic regions is all because of the expression of specific homeotic genes in these regions of the embryo. We said that there are two major homeotic gene clusters in the Drosophila. The Antennapedia, for example, activates the construction of legs on the thoracic segments, and if Antennapedia is therefore expressed in thorax, but is not expressed in the head. That is where if you now express, so in a normal development the job of Antennapedia homeotic gene is to form legs from the thoracic segments. Therefore, the Antennapedia is expressed only in the thorax but not in the head. Now, what happens if I express the Antennapedia gene in the head? This is what happens. Now, instead of antenna coming from the head, legs start sprouting from the head. So, you can see specific segment specific expression of these homeotic genes are very important for the normal development. The segments forming the head and anterior thorax are determined by the Antennapedia complex, and the Antennapedia complex is named after the dominant gain of function phenotype of its founding member Antennapedia, a mutant in which an extra pair of legs develop on the head instead of normal pair of antenna. So, these are very important genes, and that is one of the master regulators of development. Similarly, the bithorax complex. The bithorax complex is responsible for determining the posterior thorax and abdominal segment of each fly. So, the Antennapedia is primarily responsible for the head region, and the bithorax complex is responsible for determining the thorax and abdominal segment of the flies. The bithorax complex basically, the locus is a huge locus, consists of about 300 kilobase long DNA, and it controls the identity of each of the segments that contributes to the posterior 2/3 of the fly, and it consists of 3 homeotic genes. So, the development of thorax and abdomen of the fly is regulated by the bithorax complex, which consists of 3 genes, namely the ultrabithorax, abdominal A and abdominal B genes. The entire region spans to almost 300 kilobase long DNA, and the regulatory regions, the thin lines indicated here actually have been shown to be regulatory regions which are responsible for the expression of the ultrabithorax, abdominal A and abdominal B. For example, if you have mutations or deletions in this region, the expression of ultrabithorax may be affected. Similarly, if you have mutations in this region, the expression of the abdominal B may be affected. So, the dark boxes here represent the coding region, which contains the exons and introns of each of these 3 genes, and the thin line encompasses not only the coding region but also the non-coding regulatory regions, which are essential for the expression of these 3 bithorax complex genes. So, just like when you had mutations in the Antennapedia, the antenna got transformed into legs. Similarly, when you have mutations in the ultrabithorax gene, again instead of halteres coming from segment, a wings develop. So, the ultrabithorax is actually responsible for converting an haltere into a wing. So, if you now express, if you have mutation in the ultrabithorax gene, it transforms the 3rd thoracic segment into a 2nd thoracic segment. So, normally only the halteres present in the 2nd thoracic segment should give rise to wings, but if you do not have ultrabithorax, the 3rd segment also the halteres now develop into wings. So, you can see this is why the gene homeotic, these are called as homeotic transformations, where one organ can be transformed to another, and that is why the term homeotic genes have been given to these kind of master regulatory genes. Now, let us now spend some time to understand how is this Hox genes or homeobox gene expression is regulated. Now, proteins encoded by the gap and pair role genes initially regulate the expression of these homeotic genes. So, as I told, you have the maternal effect genes which activate the gap pair role genes, and is these gap and pair role genes products which are all transcription factors, and they go and bind to the promoter regions of these antennapedia or the bithorax complex genes and activate the expression of these homeotic genes. So, the initial expression of the homeotic genes requires the transcription factors encoded by the gap and pair role genes of the developing embryo. But then these gap and pair role genes are expressed only during the early embryonic development, they are not expressed in the later stages. So, then how do you make sure that these homeotic genes are expressed only in those places where they have been initiated? They should not get expressed in other regions where the gap and pair role genes did not activate them. This is done by two other important groups of transcription factors. So, at later stages proteins encoded by polycomb group and trithoracic group genes bind to these regulatory elements and regulate this expression of these genes. So, there are two important points here. The initial transcription activation of the homeotic genes is carried out or is because of the binding of transcription factors encoded by the gap and pair role genes. But gap and pair role genes are expressed only during the early embryonic development. So, in order to sustain the expression of these homeotic genes in those regions where they got activated by the gap and pair role genes, two other groups of transcription factors take over the function of gap and pair role genes during later stages of embryonic development. These are called as the polycomb group proteins and trithorax group proteins. What is important about these two proteins? The polycomb group basically act as negative regulators. Their job is to make sure that the homeotic genes which are not expressed in the regions, in those regions of embryo by the absence of the gap and pair role genes, will remain repressed. The trithorax gene are positive regulators, and their job is to make sure that the regions in which the homeotic gene transcription got activated by the gap and pair role genes continue to get expressed. So, the polycomb group of proteins act as transcription repressors. They make sure that homeotic genes are not turned on in those regions of embryo where the gap and pair role genes did not express, whereas the trithorax genes make sure that in those regions of the embryo where the gap and pair role gene products expressed, therefore activated homeotic genes, the homeotic genes continue to remain expressed in those regions. So, because of these two distinctions, the cis-acting elements to which the gap and pair role proteins bind are known as initiators, whereas the elements to which the polycomb group and the trithorax proteins bind to are known as the maintenance elements of the polycomb response elements and trithorax response elements of the homeotic genes. So, there are two important points here. To initiate transcription of the homeotic genes, you require the gap and pair rules. They go and bind to cis-acting elements or DNA elements in promoter regions known as the initiator elements. Once the transcription is initiated during a certain period of the early embryonic development, and once the gap and pair role gene products decrease or disappear, to continue to maintain their expression in those regions, the polycomb and the trithorax group of proteins take over the function of the gap and pair role genes. To sustain, therefore, to maintain the expression of the homeotic genes in these regions, these two proteins play a very important role. So, therefore, the elements to which the polycomb and the trithorax gene proteins bind are called as maintenance elements, whereas those to which the gap and pair role gene products bind are called the initiator elements. And you have two kinds of maintenance elements. One is called the polycomb response elements, to which the polycomb proteins bind, and when the polycomb group of proteins bind to this PREs, they repress the expression of homeotic genes, whereas if the trithorax proteins bind to the trithorax response elements, that results in the activation of the homeotic genes. So, I think this is the broad mechanism by which not only the expression of the homeotic genes is initiated in specific regions of the embryo, but also made sure that once the gap and parallel gene products disappear or get degraded, they continue to express in the same place by these two classes of transcription factors, namely the polycomb group of proteins and the trithorax group of proteins. So, both the polycomb group and the trithorax group of products maintain the active and inactive states of each parasegment specific cis regulatory regions by modifying chromatin structure of each regions. A lot of literature is available on these proteins, especially the polycomb group of proteins are known as the chromatin modifiers. So, they have histone methylase or histone deacetylase activity because they are involved in transcription repression, whereas the trithorax genes are primarily histone modifiers leading to activation of transcription. So, these are all chromatin modifying proteins. So, the polycomb group of proteins consists of about 40 different genes. They keep the homeotic joints of bithorax complex repressed in those segments where they have not been activated during early embryonic genesis. I think these are very important points that one has to understand if you have to appreciate how beautifully the regulation of development takes place in the developing embryo. So, you can see the homeotic gene expression is initiated by the products of gap and parallel genes in specific regions of the embryo, but once their job is over, their function is taken over by the polycomb and the trithorax group of transcription factors. So, the job of the polycomb group of proteins is to make sure that in those regions of the embryo where the gap and parallel genes did not get expressed, in those regions homeotic genes will not get activated. Therefore, they keep the chromatin in a repressed state by modifying histone, so that these homeotic genes will not get activated in those regions. The polycomb gene products form large complexes that package chromatin into a compact transcription transcription inactive conformation. So, basically they heterochromatize those regions, so that these genes homeotic genes will not get expressed in those regions. And there are basically two polycomb protein repressor complexes known as PRC1 and PRC2, and the PRC2 complex actually has a histone lysine 27 methyl transferase activity. And from our earlier studies, when we looked at the role of chromatin in gene regulation, we discussed how histone methylation and histone deacetylation play a very important role in transcription repression. So, one of the mechanisms by which at least one of the polycomb group of proteins repress the transcription of homeotic genes in those segments where they are not expressed is by methylating the histones, especially at the lysine 27 of the H3, and this histone methylation leads to heterochromatinization, and therefore homeotic genes are not expressed in this region. The H3K27 in fact is an epigenetic chromatin modification that is associated with transcription inactive chromatin. So, you can see by methylating the lysine residue of this H3, it converts the chromatin in that region into heterochromatin, and therefore the homeotic genes in this region will not get expressed. Similarly, the trithorax group of genes counteract the activity of the polycomb group of genes by maintaining the homeotic genes and their large sex regulatory regions in a transcriptionally permissive state. And all the trithorax group of gene products also have chromatin modification activities, and you can easily guess, since the job of the trithorax group of proteins to activate the expression of homeotic genes, they should invariably have chromatin remodeling activities that are known to be associated with transcription activation. It could be either histone acetylation, or it could be inhibition of histone deacetylation, or it could be other chromatin remodeling protein activities that ultimately pay off for transcription activation. So, the polycomb group of transcription factors or chromatin modifying proteins make sure that the homeotic genes are not expressed in segments where they should not be expressed, whereas the trithorax group of transcription factors make sure that the homeotic genes are expressed only in those spaces where the transcription was initiated by the gap and parallel gene products. Now, how do you study these? How do you demonstrate that or how do you map these cis acting elements of either the trithorax group of proteins or polycomb group of proteins or so on and so forth? Here is an example. For example, take the bithorax complex. I told you the dark boxes here represent the actual coding regions of these three homeotic genes, whereas the thin lines actually are the cis acting elements which are required for the expression of each of these three genes. And the bithorax gene regulation was studied using cloning the various regulatory regions upstream of a laxi reporter gene and making transgenic flies carrying these reporter gene constructs and studying the resulting patterns of expression. This is a very well known method of studying the promoter function. So, for example, if you want to study what is the cis acting region that is involved in the regulation of abdominal B homeotic gene, then you take this region of the regulatory region of corresponding to the abdominal B gene, link it to the laxi gene, Now we make a transgenic fly which is expressing the this particular gene, and now we now stain these flies with the beta galactose, look for beta galactose activity. You can see lac-ZIG expression is only those places where normally abdominal B is actually expressed, that is near the parasegment 11, clearly indicating that this region known as the IAB6 region is actually responsible for abdominal B expression in the parasegment 11. So, by using these kinds of transgenic flies expressing different regions of the cis-acting regulatory elements of this homeotic gene cluster linked to a lac-ZIG gene, the exact cis-acting elements that control the expression of these various homeotic genes have actually been mapped, and this based on this you can actually precisely identify these are the regulatory elements that are responsible for segment specific expression of these homeotic genes. So, based on these studies, specific DNA fragments that are required for initiating segment specific expression of the bithorax complex genes as well as maintaining the restriction pattern of expression and for producing segment independent cell type specific expression have been identified. So, all these cis-acting elements which are essential for segment specific expression or cell type specific expression of these homeotic genes have been identified by making these kinds of transgenic flies in which a reporter gene is linked to different regions of the regulatory regions of this huge 300 kb region, and by these kinds of study you can precisely map what are the regulatory regions which are essential for expression of these genes in a segment or cell type specific manner. So, having discussed how the homeotic gene expression is regulated or initiated by the gap and pair rule genes and then maintained by the polycomb group of proteins and the trithorax group of proteins, let us now try to very briefly understand how these homeotic genes regulate the target genes and how do they bring about the segment identity, how do they ensure that a leg comes out of a thoracic segment or an eye comes in the head or an antenna pops out of the head. Now, the binding sites, again all these homeodomain proteins are transcription factors. So, the binding sites for these Hox proteins are frequently juxtaposed to binding sites of other transcription factors like effectors of signaling pathways or proteins that determine tissue specific expression. Now, the developmental specific gene expression is pretty complicated. Now, let us not think that just the homeotic gene products alone are responsible for many of these products. Many a times the combined action of the homeotic gene products with other transcription factors are responsible for many of these homeotic transformations, because the promoter regions of many of the target genes that are involved in many of these organ development contain binding sites not only for the homeotic gene products, but also for other transcription factors which are actually activated by other signaling pathways. For example, the binding of SOX or OCT heterodimer close to the sequences bound by the HOXB1 is required for the full transcription activity detected by this sequence. So, what this tells you is that it is not just homeobox proteins alone which can do the job. They also many times have to be combined with other transcription factors, and together by the synergistic action of homeobox proteins and other transcription factors which are activated by other signaling pathways are responsible for many of the phenotypes that are responsible for this or that are attributed to these Hox genes. The Hox genes can alter the expression of ligands, receptors, intermediate elements or effectors of various signaling pathways. The functions, the Hox genes play very diverse roles. They can lead to the expression of ligands, which in turn can act as signaling molecules that can in turn activate the expression of various transcription factor. Some of the target gene products of homeotic genes can be receptors, membrane receptors to which specific ligands bind and they activate signaling pathways, or they could be effectors of many signaling pathways. Just to give an example, in Drosophila, the ultrabithorax regulates the activity of Notch, Hedgehog, DPP, wingless as well as epidermal growth factor receptor signaling pathways in the halter disc to make the halter instead of a wing. I told you ultrabithorax is actually responsible for the development of for the halters, and if you have mutation in the ultrabithorax, the halter will develop into a wing. To prevent this halter becoming a wing, the ultrabithorax has to activate a number of signaling pathways. So, the ultrabithorax transcription factor is responsible for the activation of Notch, Hedgehog, DPP, wingless, epidermal growth factor receptor. By activating all these signaling pathways, the ultrabithorax ensures that the halter remains as a halter and does not transform into a wing. Similarly, the proboscipedia homeotic gene determines the pattern differences between proboscis and the leg by modifying the Hedgehog pathway. So, this is the reason why in the previous class we studied many of the signaling pathways like the Wnt, Hedgehog, notch, and so on and so forth. So ultimately, many of the signaling pathways are actually involved in the mediation of Hox gene activities. So many of the Hox gene products, many of the homeotic gene products actually activate many of the signaling pathways, and that's how they act as transcription factors, or that's how they bring about specific phenotypic effects. Another example of this is the formation of denticles in the embryonic ventral cuticle of Drosophila embryo. The abdominal segments bear more rows of denticles, which are also bigger size than the thoracic ones. The denticles are present both in thorax and abdomen, but the denticles which are present in the abdomen are much bigger than that of the thoracic ones. This differences in the denticle size and expression is because the ultrabithorax and abdominal A activating in the abdomen a ligand for the Notch pathway called as serrate, which in turn triggers EGF receptor signaling from denticles. So the formation of denticles in the thoracic region and abdominal region is controlled by the ultrabithorax homeotic gene. How does it do it? The ultrabithorax controls the formation of these denticles in the thorax and abdomen by actually activating the expression of a gene which codes for the ligand for a Notch signaling pathway called as serrate, which in turn activates the epidermal growth factor receptor pathway. So in the thorax, the Hox genes do not activate the serrate gene expression, therefore no EGF receptor signaling is formed, and therefore the denticle bands are thinner and more slender. So you can see, when I say the denticles, which are nothing but small projections, the denticles are thinner and smaller in the thorax but bigger in the abdominal region. That is because the Hox genes do not activate, the ultrabithorax Hox gene does not activate the serrate expression in the thorax. It activates the serrate expression only in the abdomen region, and as a result the serrate now goes and binds to the Notch receptor and activates a signal transduction pathway leading to the production of epidermal growth factor receptor, which in turn is responsible for the production of the denticles on the abdominal region. So the non-production of the serrate ligand, which is the ligand for Notch in the thoracic region, is responsible for the smaller denticles in the thorax, whereas the activation of the Notch signaling pathway by the production of serrate by ultrabithorax is responsible for the production of denticles, large denticles in the abdominal region. So this is how the ultrabithorax controls differential gene expression in the thorax and the abdomen. Complex cooperative interactions between signaling pathways and homeobox genes play a very key role in the expression of target genes. For example, protein-protein interactions between the HOX-D proteins and a GLY3, a member of the sonic hedgehog signaling pathway, specify digits in the vertebrate link. We will discuss some of these things in detail as we go along. The formation of digits, for example, how a limb is formed or how a digits are formed is again controlled by a homeobox group of protein called as HOX-D, which along with another protein called GLY3, which is a member of the sonic hedgehog signaling pathway. We had discussed this in the previous class. So the relationship between Hox genes and signaling molecules is often very complicated. I gave you two or three examples of how the Hox genes cooperate with other signaling pathways, and it is these complex interactions that ultimately is responsible for the development of various structures during the embryonic development. And other important thing is the Hox genes can be both upstream as well as downstream of some of the signaling pathways and the formation of digits. That means the Hox genes can either activate the components of signaling pathway, or the components or the effector molecules arising out of signaling pathway can go and activate Hox genes. So the Hox genes can be either upstream or downstream of some of the signaling pathways. The Hox gene products may activate sonic signaling pathways like the hedgehog pathway, or the products of hedgehog signaling pathway can go and activate the homeotic gene expression. So basically what I am trying to tell you is that the regulation of homeotic genes as well as the mechanism by which these homeotic genes activate the expression of various target genes is very complicated. It varies from segment to segment, region to region, as well as in space as well as time. So by complex interactions involving various signaling molecules, various signaling pathways, other transcription factors and Hox is responsible for the ultimate development of the various embryonic structure during the embryonic development. So in the Drosophila we told that there are two major homeoblock clusters, namely the ultrabithorax and the antennae complex. When you go to vertebrates, it becomes more complicated. Many of these homeotic genes got duplicated, and therefore the vertebrate there are about 4 Hox clusters. They are localized on chromosome 7, chromosome 17, chromosome 12 and chromosome 2. and these are the various Hox cluster. There are about 38 to 40 different homeotic genes in the mouse, and these are called as Hox A cluster, Hox B, Hox C and Hox D. The vertebrate Hox genes also display collinearity. Just as in the Drosophila, we know that they are arranged in a linear fashion from head to tail fashion. The anterior genes are expressed in the anterior regions and the posterior genes are expressed in the posterior regions. So, the chromosomal localization is very well collinear with the region where they express in the embryonic development. The same way in the case of vertebrate Hox genes, there is what is called as a temporal collinearity. Genes on one end of the complex are expressed first and those on the other end are turned last. So, on a chromosome, the genes which are at the 5' end are expressed first during development, and genes which are occurring at the 3' end are expressed in the last. So, this is called as the temporal expression. That means, in time they are different. Similarly, in space also they are different. The genes which are the anteriorly expressed are more expressed much higher levels than genes which are expressed the posterior end. The reason why some of these things is that many of the anterior Hox genes are activated by morphogens such as retinoic acid. For example, if you take the Hox B cluster, there is a nice retinoic acid response element at the 5' end of the gene. Therefore, during embryonic development, some cells start producing the retinoic acid, which is a morphogen. Now, retinoic acid binds to retinoic acid receptor, that now goes and binds to retinoic acid response element and activates the expression of this Hox gene cluster. And initially, the anterior genes get activated first at high levels, and slowly the retinoic acid gradient also is slower towards the posterior region. Therefore, the posterior are less retinoic acid responsive compared to the anterior genes. So, molecules like retinoic acid play a very important role. We will discuss some of this during the later stages of this talk, and many of these molecules like retinoic acid regulate embryonic development by actually activating the expression of some of the important transcription factors such as the homeotic genes. For example, I will just give you a few examples to tell you how the Hox gene expression is very important. For example, if you have a mutation in the Hox C6 protein, you have abnormal development of the vertebral column, because the Hox C6 protein is actually expressed in specific thoracic segments during embryonic development, indicating that they are responsible for the formation of the thoracic vertebrae. And in fact, we have mutations some of the Hox C clusters. For example, we have mutations Hox C8, it can lead to development of an extra rib in the lumbar region, where normally you do not see a rib in the lumbar region, but if you have mutations in the Hox C8, it develops the extra rib. So, the function of the Hox C8 is to make sure or repress the formation of rib during in the lumbar region. So, when you have mutations, now there is no, this repression is not there, and therefore, you start forming ribs in the lumbar region as well. Just to tell you that these Hox genes play a very important role in the formation of various straddle structures. Similarly, if you have mutations in specific homeotic genes, it can lead to abnormal limb development. These are just some of the pictures that shows you, by depending upon which mutation you have in what kind of a Hox gene, you can have various kinds of malformations in the limb development. We will now take maybe limb development or eye development as an example, just to tell you how nicely the expression of transcription factors play an important role in the regulation of embryonic development. Let us take for example how the limb is formed. We know we have a limb which contains of 5 digits. Let us see how these limb digits are produced during embryonic development. Now, the first thing that happens during the limb development is the formation of what is called as a limb bud. Okay. Mesodermal somites during the embryonic development, they actually migrate to a region either in the anterior or the posterior regions to form either the legs or the hands, and the first thing that is important for the formation of these limbs, either the anterior or posterior limbs, is the formation of a limb bud. Now, in the limb bud there is a very important region, the distal tip of the limb bud, it consists of a very important structure known as the apical ectoderm ridge or the AER. Now, why is this important? If you cut off this apical ectodermal region or AER in a developing embryo, you do not get limbs, clearly indicating that this apical ectodermal region is the one that ultimately is responsible for formation of our limbs, including digits. Very interestingly, instead of removing the apical ectodermal region, if you now put an extra, if you now cut off this AER from one embryo and put it next to the apical ectodermal region in another embryo, instead of one hand, you will now develop 2 limbs, clearly indicating that this apical ectodermal region plays a very important role in limb development. If you remove this, normal limbs are not formed. If you add an extra AER, you get 2 limbs instead of 1. So, limb development, it turns out it is not just the apical ectodermal region, but the black region shown here, which is nothing but the mesoderm, which is lying beneath the apical ectodermal region. This mesoderm also is very, very important. Instead of apical ectodermal region, if you remove the mesoderm from this early limb bud, then the formation of the apical ectodermal ridge as well as limb development is suppressed, indicating that limb development involves reciprocal interactions between the apical ectodermal region and the underlying mesoderm. So, as the limb bud is formed and it starts developing into a full-fledged ledge, complex interplay takes place between the apical ectodermal region as well as the mesoderm region just below it. Now, let us try to understand what is this interaction that takes place that ensures normal limb development. It turns out, for a proper limb to be formed, your hand or legs to be formed properly, there are three axes that are required: the proximo-distal axis, anterior-posterior axis, dorsal-ventral axis. Now, let us just concentrate on one axis. How is an anterior-posterior axis formed? This anterior-posterior axis is determined by what is called as a zone of polarizing activity, or ZPA, which is located at the junction between the limb bud and the body wall. Now, if you now take this graph with ZPA, or this zone of polarizing activity, to the anterior margin of the host limb bud, it causes duplication of digits in a mirror image symmetry. What is this zone of polarizing activity? The zone of polarizing activity is nothing but these are the cells which actually produce the retinoic acid. Now, the picture when I show next, it becomes very clear. You can see here, if you take this zone of polarizing activity and then duplicate it, you get extra pair of limbs which are actually mirror image of each other. And then people actually found out this zone of polarizing activity is able to cause this kind of a limb duplication, primarily because this zone of polarizing activity actually produce cells in this region actually produce retinoic acid. So, to prove this point, what they do, you simply take a bead which is soaked in retinoic acid, and if you now implant it to the anterior margin of the early limb bud, now you can see a mirror image duplication actually takes place. So, you have 2, 3, 4, and if you now put a retinoic acid bead here, again 2, 3, 4 is formed, clearly indicating that retinoic acid acts as a powerful morphogen and is actually responsible for a developmental program involved in the formation of these 3 digits. Normally, you get only this. If you now duplicate another region of retinoic acid producing region, instead of forming one set of these 3 digits, another set of 3s are formed here. It turns out the mechanism by which this retinoic acid brings about this kind of a limb duplication is actually by activating the expression of the sonic head-shock signaling pathway. So, retinoic acid not only activates homeotic gene expression, but also activates other signaling pathways. That is why I told you embryonic development is a complex interplay of transcription factor signal molecules produced from diverse signaling pathways. This slide again shows you how the Hox gene expression plays a very, very important role during development. One of the Hox clusters in mouse called the HOXD gene plays a very important role in the limb development, and very interestingly, Hox gene expression also, these Hox gene also expressed in different regions of the developing limb. For example, the HOXD9 homeotic gene is expressed throughout the limb from top to the bottom, whereas the D10 homeotic gene is expressed only in this region, whereas D11 is expressed only in this region, D12 here, and D13 expressed only in the distal part, that is only in the digits of the developing embryo. So, this is what is called as the spatial regulation or spatial expression of the homeotic genes. That means in space the expression is different. Some are expressed throughout, some are expressed only in specific regions of the developing embryo. This is called the regulation of spatial expression of genes during development. Not only that, they are also temporally, the temporal expression also is regulated. For example, the D9 gene is expressed very early during embryonic development all over, whereas the D13 is expressed at later stages of embryonic development, that too only at the distal region. So, the expression of Hox gene is regulated both in space as well as time. So, this just tells you the D9 is expressed all over, the D10 expressed only in this region, D11 in this region, D12 here, and D13 only in the digits. And many of these Hox gene expressions are regulated by morphogens like retinoic acid. The other important thing that you have to remember about the development of limbs is that when you develop limbs, as cells are growing to form these various digits, the cells which are in between digits have to be degraded or have to be destroyed. So, a very important regulation. Again, activation and repression of transcription factors make sure that cells in between the digits undergo apoptosis, so that you get proper digits and you do not have a web. In fact, if you now take bat for example, this apoptosis does not occur in the bat, and that is why you have a web in the bat limbs, because this apoptosis does not take place there, whereas in higher vertebrates, because cells in between digits undergo apoptosis, you do not have the web and therefore you get digits. So, very beautiful regulation of gene expression takes place to make sure some of this embryonic development takes place normally. I will just give you one more example about eye development. Again, a whole bunch of transcription factors called as eye field transcription factors or EFTFs play a very important role in the normal development of eye. These eye specific transcription factors include ET, RX1, PAX6, SIX3, LH2, LSX2, TLL, optics, OPTX2 and so on and so forth. If you have mutations in any of these things, it results in the abnormal eye formation. Just to give one example, and this just tells you that again spatial and temporal expression of all these transcription factors involved in eye development plays a very important role. During day 10.5 of the mouse embryonic development, you have specific expression of the certain factors like noggin results in the formation of a neural plate. Then another transcription factor called OTX2 is expressed, and around day 11 this again leads to expression of other transcription factors, and ultimately an eye field is formed, and ultimately is results in the expression of eye. Just to give you one example of how these transcription factors are very important, if you now take the example of one of these transcription factors called PAX6, PAX6 consists of a family of tissue specific transcription factors. They are called as paired box genes. They go for a number of transcription factors, of which let us just look at what does PAX6 do. If you now duplicate the expression of the PAX6, if you now normally PAX6 is expressed in the head to form eyes in the head of the Drosophila. If you now express it in the mandible region, instead of mandibles, instead of mouth parts, you get an extra pair of eyes in the mouth. Or if you go to the vertebrate like the mouse, if you have mutations in the PAX gene, you see an embryo without eye, clearly indicating that these are all master regulators. PAX6 is one of the master regulators that controls the entire development of eye. So, I can go on and on like this. The regulation of gene expression in embryonic development is a fascinating area of modern biology, and the last 3 decades, especially after the advent of molecular biology and novel cell biology tools, excellent research has been done to understand how spatial and temporal expression of various transcription factors is responsible for the normal development of a embryo, and using model systems as Drosophila, mouse, Caenorhabditis elegans, zebrafish, a number of research groups have dissected the various molecular mechanisms by which organogenesis and embryonic cell differentiation and development takes place. And in fact, we have fairly good knowledge about how various embryonic structures are formed and what are the various transcription factors which are involved in the regulation of embryonic development. So, I have just given here a list of transcription factors, and if you have mutations in these transcription factor, what kind of abnormalities or what kind of disease phenotypes you will develop. Androgen receptor leads to androgen receptor insensitivity syndrome. If you have mutation in a transcription factor called AZF1, results in azoospermia. If you have mutations in fork head like, you get problem in results in a cleft palate as well as thyroid agenesis. Similarly, if you have mutations in the HOXD13, you get polysyndactyly, that is limb deformities. If you have mutation in the homeobox A13 homeotic gene, hand foot genital syndrome is formed, and so on and so forth. As I said, PAX2 again affects eye development. PAX3 results to different times. PAX6, we just discussed the PAX6. In fact, I forgot to mention, if you have actually mutation in PAX6, it leads to a very important disease called as aniridia. That means the iris, in the eye, the center of there we have an iris, which is nothing but is an aperture. It acts like an aperture of a camera. Now, depending upon the intensity of the light, the aperture either opens up or closes. So, if you have very bright light, the aperture shrinks, and if you have very low light, the aperture becomes large. This development of iris is actually controlled by this PAX6 gene. So, if you have mutations of PAX6, especially in a heterozygous condition, the iris is not formed. So, you have humans without iris. But if you have both alleles of the PAX6 mutated, then it is lethal. Those embryos do not survive at all, they die. So, clearly indicating that these transcription factors axis play a very important role during the eye development. What I have not discussed in this class is another very fascinating area of embryonic development, especially in plant. Just as we discussed homeotic genes in animals, there are also homeotic genes which regulate floral development in plants. So, the plant homeotic genes contain what is called as a matchbox, which is the DNA binding domain of these transcription factors, and they are responsible for floral identity. You know, the flower of a plant consists of sepals, petals, the stamen and the carpels, and the stamen and the pistil. All these four different floral organs, their expression is controlled by homeotic genes. I will not go into details of it because we do not have time. Again, if you know, for example, there are about 3 major homeotic genes that are responsible for the floral development in plants. They are called as A, B and C genes. They are all nothing but transcription factors. For example, the A homeotic gene is expressed in both sepals. During the floral development, they are expressed in both these two, first and second whorl. The B gene is expressed in the second and third whorl. The C gene is expressed in the third and fourth whorl. And if you now look at what is the importance of these genes, the A homeotic gene is required for the formation of sepals. A and B genes are required for the formation of petals. B and C is required for the formation of stamens, and C alone is required for the formation of carpels. So, again, just as we had the homeotic genes required for the various organ formation in the animals, you have homeotic genes in plants which are required for the formation of various floral organs, and if in fact have mutations in these homeotic genes, it can convert one whorl into another. For example, you can convert carpels into petals into sepals, sepals into petals. Again, homeotic transformations, just as we saw in the this is just a picture that shows how, for example, you have mutations in some of these homeotic genes, the entire flower looks green because everything has been converted into sepals. I just given some of the references for you to go and then read little bit more about how homeotic gene expression is regulated during plant development. These references can be of help. And I have also given some of the key papers. For example, this paper in 1990 by Ed Lewis, who actually got the Nobel Prize for dissecting the Drosophila embryonic development as well as homeotic genes, is a very classic paper. One must read this paper to really understand. There are a couple of reviews which gives a very important details about how differential gene expression regulates embryonic development. A few more references are listed here, and one can go through some of these things to understand how differential gene regulation plays an important role during embryonic development. I think I will stop here. I think with this now we have more or less completed how differential gene expression takes place during embryonic development. Thank you.